Welcome back, everyone. We are now starting our third and final pitch session for today. There will be more tomorrow. This session is also featuring biotech companies, and we're really looking forward to hearing from some more amazing innovators. Here today to introduce this session's companies is Dr. Lou Han, partner at Lumira Ventures. Lou is a science-driven investor who is passionate about helping entrepreneurs truly innovate best and first-in-class life sciences companies. Having started as an analyst, today Lou is a partner at Lumira, focusing on deal sourcing, due diligence, company building, and global partnerships initiatives for Lumira Portfolio Ventures. And he just recently moved into BC. So welcome, Lou. Nice to have you as part of the local ecosystem, and I will turn it over to you.
Thank you. Thank you, Wendy. Hello, everyone. It is my great pleasure to be here today. My name is Lou Han. I'm a partner at Lumira Ventures based in Vancouver office. Lumira is once again proud to support Invest in BC, an important event for the BC life science community and a unique opportunity to bring together key stakeholders in the healthcare innovation ecosystem for exciting 2 days of company pitch, panel discussion, and keynote presentations. As we listen to all the excellent presentations from innovative BC companies throughout the day today, it is important to recognize none of this is possible without all the hard work the entire Life Science BC team put in. So just want to give a quick shout out to Wendy and her colleagues working around the clock behind the scene to make this all happen. I also hope to take this opportunity to tell you a bit more about Lumira Ventures and one of the initiatives I helped to launch during the pandemic. As mentioned this morning, from the seed stage investment all the way through growth stage, Lumira have partnered with entrepreneurs in Canada and throughout North America to develop and commercialize truly transformational products that address unmet patient needs and improve the lives of patients worldwide. As Canada is the foundation of Lumira's success, we feel privileged to be an active builder of Canadian biotech and life science ecosystem. And as a function to recognize the growing need and to identify and support the development of next generation of leaders, innovators within Canadian high growth life science ecosystem, we have supported with established Lumira Fund 4, launched the Lumira Ventures Innovation Program, as we call Lumira VIP last year. The Lumira VIP is designed for select and highly qualified group of Canadian students from leading academic institutions who want to gain exposure to life science venture capital and startups are committed to pushing the futures of life science innovation forward. By working hand in hand with seasoned industry professionals and all dimensions of life science ecosystem, students will gain first-hand knowledge and insights into challenges, opportunities, processes, and people shaping the future of healthcare innovation in Canada and around the globe. I'm happy to report we just closed out our latest intake for the third cohort of the VIPs, and our team is super impressed by the quality of students applied and joined the program so far. If you are interested in learning more about the program and experience from the VIPs, I will leave a link in the chat room as well as in our booth. And lastly, as many of you know, I recently moved to Vancouver. Over the last few months, it was exciting to connect with the researchers, entrepreneurs, and investors throughout the region. And now some of you are in the audience today. As a small token of appreciation, Lumira is also sponsoring a drinks event in Vancouver on November 17th. So if you're interested in learning about this free networking event, please drop by our booth and you can find more information there. Without further ado, please join me welcome the first presenter of the section, Dr. John Coleman, President and CEO of Avivo Biomedical. John?
All right. Thank you, Lou, for the introduction, and thank you, Lumira Ventures and Life Sciences BC for the opportunity to present today. So as Lou said, my name's John Coleman. I'm the president of and CEO of Avivo Biomedical, and we're developing a proprietary enzyme-based system that can eliminate the requirement for blood type matching in organ transplant. Our founding scientists have published key proof of concept data in Nature Microbiology and Science Translational Medicine. And currently we're embarking on a $35 million Series A fundraising. And this fundraising is sufficient to get us through clinical proof of concept in organ transplant. Organ transplantation is a life-saving procedure. And last year, there were over 41,000 organ transplants in the United States. Yet, the demand for organ transplants and organs greatly outstrips the supply of organ donors. Currently, there's over 116,000 patients on transplant waiting lists. And this number only reflects a fraction of the true demand, where there's hundreds of thousands of additional patients who would benefit from an organ transplant. Yet, with this huge need, there's still a massive underutilization of donor organs. Last year alone, there were over 28,000 potential viable donor organs that were not retrieved from willing donors. And most of these organs were lungs and hearts. In addition, there were over 8,000 organs that were retrieved from donors and not transplanted. The first step in the donor allocation process is to match blood group. And the transplant for different blood groups varies, with Group O patients waiting the longest and their waiting lists moving the slowest. Group O is also overrepresented on all the waiting lists. And coincidentally, Group A is overrepresented amongst the organ donors. And our technology is able to overcome the blood group incompatibility in organ transplantation. And we do this by converting the blood group antigens, which are on the cell surface of cells, and we convert them to the universal donor antigens. And we do this by removal of the terminal sugar off of these antigens using our enzymes, cleave that off, and it converts it to the universal blood group. And these organs can then be put into any patient. So the way this would work in practice is you have an A donor, the organ is retrieved, it is placed into an organ perfusion device. Our enzymes are added to the device and the organ perfusion solution that's pumping through the organ. During the transportation phase, the organ is converted from A type to O type, and then immediately prior to transplantation, our enzymes are rinsed out with clean perfusate, and then the organ put into the patient. So this is a completely ex vivo process, and it is being designed to be integrated into the standard organ procurement workflows and be compatible with all the organ perfusion devices on the market and organ perfusion solutions that are on the market. Working with our collaborators at the University Health Network in Toronto, we've demonstrated key proof of concept data converting human lungs from type A to type H. In this study, paired lungs were divided into control and treatment arms, and we can see in the graph on the right that we're able to forward over a 97% reduction in the antigen expression levels on the epithelial of the lung tissue. And when these lungs were challenged with O plasma, which contains antibodies to A and B antigens, we can see in the red line above that the treated lungs did not bind any of the antibodies and had no immune response when compared with the control untreated lungs. We believe that this product has a potential blockbuster opportunity. And looking at the discarded organs and the unretrieved organs, in addition to accessing living donors, we believe there's the potential to increase the number of transplants per year by over 16,000, representing approximately $2.5 billion opportunity in the United States alone for type A organs. And when this is expanded to include B and AB organs and the rest of the global market, it's truly a huge opportunity. We're currently progressing our organ donor product through preclinical studies and plan to file an IDE, or an investigative device exemption application, in 2024. And this is progressing along the FDA's PMA device pathway. And we're proposing that our first clinical study, although we're not quite ready to start clinical studies yet, but we're proposing our first clinical study would be in living kidney donor transplant, where we want to demonstrate safety and efficacy. Our intellectual property portfolio currently consists of patent applications that we've licensed from the University of British Columbia, which are our national phase filings around the world. In addition, we have access to improvements made by our UBC founding researchers. In particular, they're working on our technology applied to blood products, and this work is supported by a $3.5 million CRADA grant with the U.S. Armed Forces. So this Series A financing that we've just initiated is sufficient funding to complete our first clinical study in organ transplantation. The company was formed in 2020 as Aviozymes Biomedical as a spin-out of UBC. We changed the name in September of this year to Vivo Biomedical. We currently have 12 FTEs and we've raised $3 million to date, in addition to $4 million in grant funding. And we're currently raising $35 million to progress our lead product through to clinical proof of concept in humans. Thank you for your attention, and I'm happy to answer any questions through the chat, or you can contact me via email, or John Barkley, who is in the chat room as well. We're happy to answer questions. Thank you very much.
Thank you. Thank you, John. Interesting technology going after a serious problem. Coming up next, we're going to hear from Dr. Mohammed Gamal, CEO of the Gelmar company. Mo, take it away.
Thanks, Lou, for the introduction. Hi, everyone. This is Mohammed. I'm going to talk today about our proprietary technology, which is our biomaterial, Gelmar. Where the company is named after, the Gelmar company. So what is Gelmar? Gelmar is an abbreviation of a more complicated chemical name, so it's not important. And it's a semi-synthetic hydrogel biomaterial. So it's first mentioned in the year of 2000s. It has been widely used for more than 20,000 research papers. It's a wide range of applications. It has 3D cell cultures. It has been used a lot in 3D bioprinting, microfluidics, tissue engineering, and a lot more. So that's the material itself. What is our technology? Our technology is a new way to produce this Gelmar. I want to tell that this Gelmar is really difficult to produce and very expensive to purchase, and it's hindering a lot of good research that can go on. It's inaccessible to buy. There is a lot of problem in the supply. It's very expensive with supply issues as told. It's inefficient to make. It takes around 2 weeks to do 5 grams of that material. The process varies from a lab to another, so the results may be inconsistent and irreproducible. A lot of problems technically, so that makes this material, which is really useful, inaccessible. So it's a massive challenge to the advancement research. So the current potential for Gelmar in general is in bioprinting market. It's around $1.7 billion to be in 2024. It can be used in cell culture protein surface coating with around $660 million by 2026, and in the pharmaceutical and cosmetic toxicology testing, which is a huge market, like around $14 billion. So our solution is we have patented a new process to produce this valuable material in a much more efficient way. You have a quick production time. We have guaranteed sterility, which is also a problem, like in the biological labs, if the product is not sterile, which is the case currently with in-house preparation of that material. We have customizable properties. We can customize it to the requirement of any research group, and this would highly increase the research efficiency. Our material has no requirement for regulatory approval because it's a research material. So our team is myself. I'm CEO and founder of Gelmar Biotechnologies. This was a patent we developed at UBC during my years of study. Dr. Ian Foles, he's our collaborator doing more R&D research on how to use this on a high scale for other applications. For sure, you all know Derek Gradzi. He's my mentor through EITBCO incubator program, and he continues like helping and providing a lot of advice. Hannah is our market development, Saidul is for production, Christine is our marketer. Achievement to date, we have patented our protection in Canada and the US. The patent is already guaranteed and allowed in Canada, and we're waiting just for the US to follow. Legalization, and we have done all our legalization and have invested personally in the lab equipment for about $40,000. We have a lot of partnerships with MyTax for the R&D we are undergoing. IRAP was really helpful with a lot of support, and we are still looking for more from IRAP. Digital Skills for Youth helped us with initial hirings. The UBC UILO was really helpful in finalizing the patency and the license and service agreements for using a lot of UBC facilities. Our academic collaborator, Dr. Ian Folds, is really helpful in building our vision for the future. And we are currently working from UBCO, where our lab is there for our manufacturing. Our achievement to date in terms of sales, we have year-to-date sales of more than $500,000. Plus, we have guaranteed a governmental contract for 3 years for Ocagel, our material. Distribution channels, we use like all possibility of connections, social media, and our website. And we're doing networking and conferences like this one and the others. Projected revenues are like expecting $600,000 this year. We're actually a bit higher than the expected this year for our revenues. Year 2023, we expect $2 million next year, and by 2027, we plan to dominate the market of the JALMA, which is more than $25 million as our contribution. So we have 3 years direct by contract to the government of Canada at the moment. We have partnerships with JALMA researchers worldwide, and this would be our main growth drivers. And we are going after scientific supply and equipment companies. We found out that some of the already present JALMA companies are buying from our JALMA to supply their clients. So we're mostly dominating the market soon. So we're seeking $2.5 million for 10% equity of the company. The use of fund will be mainly of expansion of our manufacturing facility, increasing marketing efforts, and continuing IP protection in the key markets. Our mission is to increase academic and drug research efficiencies worldwide with our patented, high quality, and fairly traded Ocagel. Thank you.
Thank you, Mo. Great. Thanks for taking the time to walk us through your company. Coming up next, let's welcome Dr. Carolina Lallet. She's going to tell us all about the VoxCell bioinnovation. Carolina.
Thank you, Lou. I appreciate the introduction. Hello, everyone. When I was 6 years old, I remember seeing my mom going through breast cancer. And although at that point, I don't think I knew what breast cancer was, I knew exactly what pain was. And a few years later, unfortunately, that breast cancer came back. And at that point, I was doing my PhD, and I decided to dedicate my life to trying to understand what makes cancer so resistant, and how do oncology drugs actually get to the market through the drug development process. And to my surprise, I realized that actually 95% of oncology drugs, they fail to get to the market. And this high failure rate is associated to how these drugs are tested. They are tested in very simplistic platforms in the lab, and then they go to animal models. And then, of course, that goes without saying, animals are not people. And then they are tested in people. What we see is the correlation between lab data and clinical trial is actually less than 8%. So we are talking about an industry that is spending $500 million just in preclinical trial per drug. So what I thought was, can I create something artificially that would allow pharmaceutical companies to test anti-cancer drugs in a meaningful way? And that's how VoxCell was born. At VoxCell, we are using bioprinting technology to create what I like to call as artificial biopsy samples. So here's an image of our tissue models. And as you can see, it's 3D bioprinted and it contains blood vessels. So what we see is the result is a human-like cancer tissue models. The pharmaceutical companies can then inject the drug candidates through the vasculature and analyze how this drug distributes throughout the tissue in a very human-like way. There has never been a better moment to put this technology on the market than right now. The FDA Modernization Act of 2021 was a bill proposed in October 2021, and that has actually just passed in the end of September this year. And for the first time, the FDA is trying to find alternatives to animal tests. And also for the first time, it has been referred as one of the alternatives to be using bioprinted model. So this is great for Voxel, and this is the right moment for us to be putting our technologies on the market. Because we're going to be using our tissue models as a screening platform to screen drugs to see which drugs have a higher potential to work in humans, we actually do not require FDA approval. And we actually spend a lot of time talking to potential partners. So as you can see, the big names in here, our idea of talking to them, we're actually asking them what they want to see to adopt our technology. So our validation studies of our tissue models have been designed according to their feedback, and it's going to be ready in the beginning of next year. We have also conducted a market assessment, and we have received very positive feedback for our product profiles. And this market assessment has been with support of IREP, and we just got into CDL. Voxel is creating these tissue models by combining bioprinting techniques, and we have created our first, it's Canada's first high-resolution bioprinter, and we combined that with proprietary bioinks. What that means is with the printer's cartridge. And it's similar to the printer you have at home. You buy a cartridge to plug in your printer. That's what we created, our proprietary bioinks. And we also design our own vasculature generation software. So this software is what is responsible for creating our tissues vasculature and allowing us to create these human-like tissue models. What we will be selling is the consumables. First is our vascularized cancer tissue models. That is the outcome of our printing process. And we plan to sell this directly to consumers in a very easy plug and play situation in which people will be able to inject the drugs directly through the blood vessel. We also plan on selling our inks because our inks, even though they were created primarily to use internal, what we saw is we made something that is compatible with any other bioprinter on the market. So people buying our inks can plug in their printers. However, they will not be able to replicate the same tissue models that we are selling because they will not have access to our printer or our software. We are going to be keeping in-house. In terms of our tissue models, what we see as competition is other preclinical testing techniques out there. However, none of them display the complexity of what we are creating. We are really targeting and marketing ourselves towards being a tissue engineering company, providing reliable preclinical testing platforms for drug testing. In terms of the bioinks, what we see is other bioink companies out there. However, as I mentioned, Voxel created something that is truly universal and can be coupled with any other bioprinter. On top of that, our ink is tissue specific. So you're no longer buying a collagen-based ink or a gelma-based ink or a gelatin-based ink for bioprinting. You are buying something that is really breast tissue or lung tissue, and it's really ready to go. We also have extended shelf life with our ink. Our IP strategy is to create an IP portfolio in bioinks, tissue models, software, and hardware. Our software will be kept as a trade secret, and our hardware, we right now are updating our printer configurations to be able to print 1,000 tissue models a month and be able to generate $4 million in revenue per printer. Our market is the bioprinting market that is separated in 3D models and bioink, with North America dominating 30% of it. Our customers will be academia, research centers, contract research organizations, and pharmaceutical companies. What we see as academia and research centers, the focus is really on the bioinks, while CROs and pharma, the focus is on the tissue models. And we have a 70% margin on the tissue models and a 90% margin on our inks. I'm Dr. Carolina Valente. I have a background in tissue engineering, and I'm the CEO and CSO of Voxel. Dan has joined us as our CBO. He is a serial entrepreneur, has exited many companies, and is working with me on the business development. Dr. Steiner has just left Aspect and has joined us full-time to develop our printer. And Dr. Voss is leading our validation studies of our tissue model. We are 12 full-time employees, and we have combined expertise in everything that we need to succeed with this company. And on top of that, we have a very strong group of advisors from pharma to oncology to optics and amazing board members. Voxel opened our pre-seed round this year, and our goal was to raise 1.3 million Canadian dollars at a price round. And we ended up getting oversubscribed. So we now are extending our round to 2 million. So we are seeking another 350,000 by the end of the year. And our idea is to put these tissue models on the market in the beginning of next year. And our bioinks are going to be going to the market in the end of this month. So for the investors here in the room, we have a data room ready, and we also have very comprehensive due diligence report that I can put available to you. Our mission and vision, and the mission being the most important part, is to accelerate life-saving therapies, getting to the cancer patients sooner. Because we know the drug development process is costly and is inefficient. But the FDA is trying to change that, coming from with a shift from the animal models. And that's exactly what we are doing at Voxel. We are creating human-like cancer tissue models and universal bioinks. Thank you very much.
Thanks, everyone. Lou, you coming back?
Yeah, I just gotta say thank you to Carolina for a great presentation, but over to you.
All right, thank you. I was going to say the same. Great presentation, Carolina. Your passion is infectious. So thank you to all of our speakers today. Lots of really interesting innovation there, and really three quite different things that you're working on. So nice to see all of that, and I hope you make some meaningful connections through these pitches. So with that, I will be wrapping up today. We've seen a variety of life sciences innovation presented today, from companies who have innovative platforms to support drug discovery or to speed up drug discovery and clinical trials, to early stage companies advancing their scientific discovery and ultimately developing life sciences innovative solutions that are destined to have an immeasurable impact on cancer, autoimmune diseases, epilepsy, mental health, transplants, and more. It is so inspiring what you're all working on, and thank you for taking the time to share with us today. As we look ahead and comprehend the impact of the pandemic, we know that we need to drive both economic recovery and competitiveness while ensuring the sustainability and reliability of our healthcare system. Clearly, the life sciences sector is critical and well-positioned to address these issues. In BC, as we've heard today, we have a strong pipeline of innovations that will play a significant role in achieving those goals. And it's an incredibly strong group of talented, scientifically brilliant people. We're so blessed that you've chosen BC to be where you do this work. On that note, I'd like to thank everyone for joining us today. In addition to hearing the pitches and panels, I hope you take the opportunity to connect with each other to explore collaborations, investment options, and networks. I'd like to thank our remarkable sponsors who are so critical to putting on an event like this. Our presenting sponsor, Lumira Ventures, our conference partner, Admare Bio Innovations, our session sponsors, AstraZeneca Entrepreneurship at UBC, Genome BC, Gowling WLG, Global Affairs Canada, Nimbus Synergies, and Novature Ventures. Our keynote sponsors, the British Columbia Securities Commission and EY. Our event sponsor, Innovative Medicines Canada. Our networking sponsor, Gowling WLG. Our technology sponsor, Owen Wiggs, and our event supporters, Berkeley Canada, the BC Ministry of Jobs, Economic Recovery and Innovation, and Merck. I'd also like to thank our leadership volunteers who I referenced earlier today. We have a group of people that generously give their time to review the applications to select who can present here. We had almost 80 applications and selected 30 companies, so that's a big amount of work for them, so thank you. We also have some leadership volunteers who all of our presenting companies would have met, who do pitch prep in advance of the session today. So thank you for that. We really appreciate the time that you give to our sector. Finally, a big thank you to Ryan, LSBC's Director of Partnerships and Events, and whom we refer to as our production manager when we're doing these online events. Claire, who's been working tirelessly to ensure our speakers are prepared and the event goes off without a hitch. And in addition, Joanna, who recently joined LSBC in a marketing and communications role, who has been very active both before the event and today, ensuring that we are sharing what we're learning and seeing today on social media. A reminder that the virtual event platform remains open throughout the 2 days, including the expo hall, and 30 days after, so you can go back and not only make the connections, but view the sessions again. We'll reconvene tomorrow at 9 a.m. Pacific Standard Time, where the format will be very similar to today with keynote speakers, pitches, and panels. We really hope that you'll join us again tomorrow and engage again. We'll see. We're moving from biotech and therapeutics to medtech and virtual health companies for the pitching, and we have some exciting panels on the state of the venture capital world and investment, and Global Affairs Canada will also be coming to join us. So with that, please remember, if you post about our conference on socials, to tag us at #LSBCInvest and use our handle LifeSciencesBC under LifeSciences_BC. Have a great evening, everyone, and I look forward to seeing you again tomorrow.